Okay, good evening, everybody. My name is Nicole Gordon and I'm the CEO of Genetics. Welcome to the latest lecture in our Diving into a Gene Pool series. Tonight's talk is called CRISPR Cas9, the future versus the slippery slope. We have an absolutely fantastic lineup of speakers this evening, so I'm sure we're in for a real treat. Firstly, just to let you know that the session is being recorded, and secondly, to ask you if you do have questions as the speakers are presenting, could you please put them in the chat and we will get to them at the end of the session. Genetics was founded back in 2009. The aim of Genetics is to raise awareness, educate, and test for Jewish genetic disorders. JGD is a term that we often use to describe genetic conditions that are more common in people of Jewish ancestry, including people from Ashkenazi, Sephardi, and Mizrahi backgrounds. Genetics tests for 47 of the most severe JGDs. These are all recessive conditions, all of which have no known cure. Since 2014, we have screened almost 6,500 individuals, educated thousands of young people, and identified numerous carrier couples who we have helped go on to have healthy families. Recently, we have been appointed by NHS England as the official engagement partner of the Jewish BRCA testing programme, working alongside High Cancer Care and hundreds of stakeholders, both inside and outside the community. Since launching the programme properly just over a month ago, 17,500 people have signed up to be tested for these faulty genes. BRCA gene faults are more common in those with Jewish ancestry compared to the general population. 1 in 40 Ashkenazi Jews and 1 in 40 Sephardi Jews versus 1 in 250 of the general population have a BRCA gene fault. As the engagement partner, Genetics' role in this programme is to ensure that people understand the implications of taking the BRCA test and ensuring that screening participants are supported through every stage of their BRCA journey. This test gives people the knowledge and the power to make an informed choice for theirs and their family's future health. For more information about either of our tests, please visit genetics.org. I would now like to introduce Dr. Juliet Harris, Trustee of Genetics, who in turn will introduce our guest speakers. Juliet is a specialist genetic counsellor at Northwest Thames Regional Genetic Service. She was in the past the dedicated NHS genetic counsellor for genetics. Juliet was particularly keen to be involved in this evening as her own PhD at Imperial College was looking at the potential for gene therapy to be used in muscular disorders. However, she told me it was very tough, as the technology to make it work simply didn't exist back in the 1990s. Juliet is very excited at the advances that mean that gene therapy is now a reality. Over to you, Juliet.
Thank you, Nicole. At Genetics, we firmly believe that knowledge is power. And today we are looking at the power of genetics to shape our future. As Nicole mentioned, up until now, all the recessive genetic disorders genetics screens for have no known cure. But with modern gene technology, this could be about to change. Indeed, one of the diseases we screen for, spinal muscular atrophy, is one of the first diseases in the world to be treated using gene therapy. So gene therapy is now a reality. CRISPR Cas9 is the name of the technology that is leading the way on many of the gene therapy breakthroughs, with the potential to treat conditions from cancer to infertility to even the muscular disorders that I attempted to work on for my PhD. It is an amazing technology that can switch off and correct faulty genes with the real potential to cure disease. But as you can imagine, the technology that allows us to rewrite our genetic code, however exciting, is fraught with ethical challenges. Tonight's event is called CRISPR Cas9, the future versus the slippery slope, and we are extremely honoured to have with us 3 amazing speakers. Dr. Soren Hough is an American-born science communicator and researcher. His scientific career has spanned institutions in the US, in Canada, and the University of Cambridge. Soren moved to London to work as the head science writer at a CRISPR and AI biotech startup called Desktop Genetics in 2016. During his PhD at Cambridge, He studied how the cell responds to DNA damage and discovered a rare developmental disorder. He has spoken about the ethics of CRISPR gene therapy in podcasts, in lectures, and in the popular press, always with an eye towards engaging the wider community. Dr. Ajetenmobi oversees venture programs at Pioneer Group, which brings together life science experts, executive talent, venture capital, and lab infrastructure to help funders fund and scale breakthrough life sciences startups. He leads a team that has supported over 100 companies, which have raised in excess of £350 million in funding. Ayokumni was previously head of sequencing services also at Desktop Genetics. He has a PhD in clinical medicine from Trinity College, Dublin. Soren and Ayokumni have worked closely together and in 2017 published a textbook chapter on the ethics of heritable genome editing, as well as other publications. Our third speaker is Rabbi Kenigsberg, who is the rabbi of Beit Knesset Hanassi in Jerusalem. He served as a visiting rabbi for Mizrahi UK in London for 4 years, where he was the rabbi of Magen Avot Synagogue and Bnei Akiva, UK. He is a graduate of Manhigut Toranit, a prestigious 5-year intensive program training rabbis and leaders for the Jewish world. He holds a master's degree from Bar-Ilan University in Science and Halacha, Jewish law, and has lectured and published internationally on topics relating to the interface between Jewish law and modern technology. We are so delighted that they are all able to join us tonight. I am now handing over to Dr. Hough and Dr. Ajetenmobi to tell us their perspective on the use of CRISPR Cas9. Over to you.
Great, thank you so much. Let me just get this screen sharing. All right, and get started. All right, well, thank you so much for that. And thank you so much to Genetics for having us come and speak today. I know I speak for AJ and myself when I say that it's a real honor to be here and have a chance to interact with a new community in the context of CRISPR and the ethical considerations therein. So, as was mentioned before, this event is about CRISPR and its potential as well as its potential downsides and the ethics. The ethical questions that CRISPR can raise. And the reason I use CRISPR here, I just want to clarify for anyone who might be wondering why I've said CRISPR, not CRISPR Cas9. CRISPR, I think, more generally captures the range of tools that fall under a similar umbrella, along with Cas9, which is just one of these. So I think the first question we want to ask here, and what you can see in this little looping animation we've got, is an animation, but it depicts more or less what happens with CRISPR. So you have DNA across the middle there, and it's being actually broken by Cas9, which is the big purple blob that's binding to the DNA. And what CRISPR actually is, and I think it's really interesting to think about where it came from, was a sort of bacterial immune system. So the way to think about this is that, bacteria, like humans, can be infected with viruses. And when they're infected with viruses, that can be detrimental to the bacterium. And so some bacteria developed a sort of immune system as a response to this, where by stealing a little bit of the viral DNA that was present after the infection with the virus, the bacterium could then recognize the next time it gets infected with that same virus and say, hey, that's the same virus that infected me before. And what it will do in response to that is chop up that DNA from that virus and prevent it from continuing its infection. So it's a sort of response, this direct response to viral infections. And I think it's useful to think about it that way, in a kind of similar way to our immune system, although obviously the mechanisms are quite different. But the great innovation of CRISPR was, okay, great. So this is how it works in bacteria, but what if we could take it out of bacteria and have it work as a molecular scissors? And then I think is well depicted here. and direct CRISPR to cut DNA in other contexts. So not just in the context of a bacterial immune system, but what if we could cut DNA in other cells, for example, in human cells or mammalian cells? And so that's what CRISPR is. And I think it's helpful to think of CRISPR essentially as little molecular scissors that can cut DNA and ultimately, with more recent advances, start editing DNA. So not just breaking it, but rewriting it. Kind of like if you think about a Word document, you can not only delete letters or separate lines, but you can actually replace the letters with new ones and maybe form new words, which we would call genes in this case. So that's a really interesting sort of, and I think your mind starts to race with what's sort of possible there. And I think it's also worth, crediting the people who are responsible for bringing this out of the realm of bacteria and into the realm of editing in other contexts. And so these two scientists won the Nobel Prize in 2020 for their original paper in 2012, which is something that AJ will talk about in just a moment. By using CRISPR, again, in this context of cutting DNA, in their case, in a test tube, and saying, actually, we can use this as a directed scissors to cut DNA wherever we want. And this new ability to do that has had a myriad downstream consequences and has really facilitated research and medical development. But I'll turn it over to AJ now for a little bit of history on the past decade of CRISPR research.
Thanks, Soren. So I guess one of the most remarkable elements to CRISPR is just how quickly it developed and matured as a technology, as this timeline illustrates. It's funny how quite commonplace it's become in many aspects of scientific research, and only 12 or so years after initial discovery, we're already seeing real-world clinical applications. So all of this began, as Soren mentioned, in 2012, when it was discovered by Jennifer Doudna at UC Berkeley and Emmanuel Charpentier, when it was first used to edit DNA in a test tube. Then a race began to see who could edit DNA in mammalian cells, which I guess would be the next major frontier in the development of this technology. And this race was won by researchers in MIT, led by Feng Zhang in 2013. And incidentally, 2013 was the year that I was first introduced to CRISPR. It was during my PhD, and I remember thinking that, well, A, this is incredibly confusing, and B, CRISPR is quite a silly name. But on top of that, at the same time, I also felt that I was reading about something that was potentially momentous and game-changing. Created a real sort of buzz and excitement around something that I had next to no knowledge about at the time. And from that moment on, I devoured every piece of news I could about CRISPR, which was fortunate because its impact only grew from then on. In 2014, the first disease was corrected using CRISPR in an animal model. And Soren, I believe that was around the time you were introduced to CRISPR.
Yeah, so I actually I had graduated. I also graduated from undergrad in 2014, and actually the person who corrected a disease in an adult mammal, this was in a mouse, it was a liver disease, became my boss. I applied to work in his lab. Wen Zhua, who works at UMass Medical School, University of Massachusetts. And he basically introduced me to CRISPR. And I remember my interview with his lab where he was telling me about, this new thing that let us, actually edit DNA, not just, in a tube, but in cells and potentially in organisms. And his claim to fame was this big paper, which showed that you can actually use it as a medical intervention. So that's how I got introduced to CRISPR. And to be honest, at the time, I thought it was science fiction. And it's been,
Likewise.
It's been about 10 years since then, and it still feels like science fiction.
Yeah, couldn't agree more. And then on to 2016, where we, I think, start to see the double edge of this rapid technological development. So on the one hand, bypassing regulations to bring CRISPR edited mushrooms to the market, and then on the other hand, working within regulations to bring a new cancer therapeutic to clinical trials. 2016 was also interesting for other reasons, which I'll get into on the next slide. But sticking to our timeline, this dichotomy, I guess, of working both within and outside regulatory frameworks became even more important in 2018 when the first human babies were edited with CRISPR, a topic we'll return to in greater depth later on in this presentation. In addition, we had the first CRISPR edited foods being available for human consumption. And I guess to cap off this remarkable rise in CRISPR development, merely 8 years after their initial paper, Jennifer Doudna and Emmanuel Charpentier were awarded the Nobel Prize in chemistry for their CRISPR discovery, which is really an unprecedented race of return from discovery to Nobel Prize. Now, I mentioned previously that 2016 was an interesting year for other reasons. And this picture perhaps illustrates why. Perhaps not in the grand story of CRISPR, but certainly to Soren and I personally, 2016 was the year that we first met. And it was the year that we both joined Desktop Genetics, which was a startup developing algorithms to help scientists design better CRISPR experiments. And if you look through the photo, you might see a couple of familiar, if perhaps much younger, faces in the photo. But perhaps our most prominent achievement while working at Desktop Genetics was our work with Professor Wasim Kasim of UCL and Great Ormond Street Hospital, supporting the development of new therapies against childhood leukemia. And without doubt, the most impactful aspect of my career to date is in the knowledge that there are children walking around today who would not be there without the therapies that we helped to develop.
Yeah, and I think it's hard. Well, first of all, I'm very jealous that you got to be on this paper. Unfortunately, I helped support it from the background, but and I'm sure many more people were involved in this from Desktop Genetics who couldn't all be listed as authors. But it's really remarkable because I think it's important to just get the punchline of this, which is that a cell therapy developed with CRISPR and using quality control that was done at the company allowed I think it was three children to beat leukemia, cure cancer. I mean, that's what we're talking about when we talk about the excitement around what CRISPR can do. That there's this, these intractable and definitely life-threatening conditions can be treated with CRISPR, and that I think brings us to the landscape as we see it today. So that was in 2017. We won't go through this in any great detail, but just to emphasize, every little colorful dot and square and triangle you see here is in fact a CRISPR therapy that's being developed. And in fact, this is a year out of date, or almost a year out of date. This is June 28th, 2023. And this was produced by somebody on Twitter named Andrew. But the point being that there's just a huge landscape now for CRISPR therapies. And even since this came out, as I mentioned, this is already out of date. Like some of these therapies have advanced all the way to the clinic. So I'll turn it over to AJ to talk a little bit about that.
Yeah, since that graphic was created, we've seen the first approval of a CRISPR therapy in the UK, which is quite remarkable. In fact, late last year, 3 prominent regulatory bodies, the European Medicines Agency, or EMA, the US Food and Drug Administration, FDA, and the UK's regulatory body, the MHRA, all approved the use of CRISPR therapies for 2 debilitating blood disorders known as beta thalassemia and sickle cell anemia. Additionally, we're seeing late-stage clinical trials showing promising results for treating a swelling disorder known as hereditary angioedema, as well as clinical trials for treatment of a disease of the heart and nervous system known as ATTR amyloidosis. So we're really seeing this as the previous graphic shows and this image shows, this explosion in the growth and development of this technology for treating previously intractable disorders. However, while there is a lot of justified excitement about the promise of CRISPR in treating devastating and deadly diseases, there's a lot of controversy, not least highlighted by what happened in 2018, the year of the CRISPR babies.
Right. So I think it's, I think it's very easy with CRISPR to sort of scaremonger about the things that could go wrong and where those ethical boundaries might be breached. But I do want to just say, modern genetic research and biological research, especially the kind that I was doing, for example, in my PhD, wouldn't be possible without CRISPR. So, it really has transformed, accelerated, and made possible things that before were virtually impossible, as well as these therapeutic developments. But as AJ just mentioned, in 2018, this announcement was pretty remarkable, which is that this scientist, Dr. He, who worked in China, announced that he had edited two, and it turns out actually there were three babies. And essentially what he was trying to do in this case, at least his claim of what he was trying to do, you can come to your own conclusions about what was really going on here, was to edit these babies to remove a cell receptor, which would essentially make them immune to HIV, which on its face sounds like a great thing, right? Immunity to HIV would be really exciting. And this is something that actually a certain amount of the population already has. They have a mutation in this cell receptor called CCR5, and they are actually naturally immune to HIV. And so he's essentially taking advantage of this natural mutation to imbue this on the children. But we have an interesting question here, which is, first of all, we have a lot of we have a variety of opportunities to prevent transmission, for example, from mother to child if they have HIV already. We have mechanisms of managing HIV, and we have obviously mechanisms of preventing acquiring HIV in the first place in other contexts. And I think there was a real question on what the advantage was of using this new technology, which on one hand can be very exciting, but on the other hand has its own risks associated with it, right? There are things about HIV which we don't fully, about CRISPR, which we don't fully understand yet, right? There are specificity issues where we might edit where we don't intend to, or where there may be an immune response or other issues that we aren't fully able to predict. And then also, what we're targeting is also a question, right? CCR5, we know if you don't have it, you're immune to HIV, but we also don't necessarily know what other roles CCR5 might be playing in human biology. And so to take that leap and say, okay, well, we should just do this because it would have this one effect, we don't know what the sort of knock-on effect would be. And finally, I think it's important to just think about the disaggregation here of, on one hand, if you edit a cell, take a cell from a patient, edit it and put it back, which is what happened with the leukemia patients. That's one question. But a very different question is when you edit directly in a person, or even more dramatically, if you edit in an embryo or in the gametes and sex cells, you have the potential of not only changing that person, but also their subsequent generations. So it's a much thornier question than perhaps it would be in the context of a cancer therapy. And I think it's important to also think about how the scientific community responded, which has been a huge influence on why AJ and I have been so involved in this discussion as well.
Yeah, I mean, from my perspective, almost as controversial as what He Jiankui did was the response of the scientific community. This photo here is a snapshot of a conference that was scheduled, the second International Summit on Human Genome Editing, where the organizers knew that Dr. Hay was going to present certainly what I consider, and I think Soren would agree with me, consider to be unethical research on the editing of human babies. Initially, he was to present on another topic, but then he subsequently informed the organizers that he was instead going to present this work, and the organizers decided to platform him and to put him on the agenda, and by doing so to lend him legitimacy. And I suppose we can all form an opinion on their culpability in that scenario.
Yeah. And just to emphasize the point on this, because I think it's worth knowing what was going on in the scientific world at the time. In 2017, there were a huge number of articles published by leading the top scientists in the world saying that we should draw a red line around this kind of research. So there was a very clear statement from the scientific community. But then when push came to shove, there was a real question of whether or not that was backed up by any sort of real commitment to this stance. And there were dissenters who didn't think that that position was reasonable in the first place, but it did raise this really fundamental question. But ultimately, when you have Dr. Hay flanked by two leading scientists and the a variety of the world's leading scientific organizations, emblazoned across the top of the screen, it really, I think it makes a bit of a statement, as AJ said, it lends an air of legitimacy. But if anybody's curious, this video is on YouTube. You can watch the whole thing. You can see his whole presentation.
So this CRISPR baby story, I think, really gets to the heart of the debate around CRISPR therapies. And we find it helpful to view the development of these therapies within three buckets, which we'll walk you through right now. And in the first bucket, we have what we consider to be life-saving interventions. So this is to treat disorders that are fatal or highly debilitating, disorders such as Tay-Sachs, which is prominent disproportionately so within the Jewish community. Cancer, I'd mentioned childhood leukemia earlier. DMD stands for Duchenne's muscular dystrophy, which is a degenerative muscle disorder, and cystic fibrosis, which is a degenerative lung disease. So the critical factor here is that CRISPR, the application of CRISPR technology for therapeutic intervention will directly save lives. So, the prominent safety risks, Soren had mentioned the specificity of CRISPR and trying to prevent anything happening off target. So any edits happening outside where it's intended, that is a significant safety risk. But the opportunity to save lives supersedes those safety risks within the regulatory frameworks that we've developed. So that's bucket one. That's I guess really well articulated and defined. And then in bucket three, I think we would also find ourselves looking at areas that are well defined. So bucket three would be more cosmetic interventions. So we would be looking at interventions that modify aesthetic preferences, things like eye color or baldness, although some people suffering from baldness might say that is a debilitating condition. Things like behavioral modification or even athletic performance. So myostatin is a protein present in muscle and can be altered to build muscle, for example. But critically, these interventions are not life-saving, right? So they are ultimately preferential, and the application of CRISPR technology within these contexts would be deeply ethically questionable. And I think most people would sort of agree with that. I guess it's in the middle bucket where we might find more nuanced debate, and Soren can sort of walk us through some of that.
Yeah, so I mean, just to emphasize that point that, bucket one and bucket three are just such clear, for me, they're such clear, whether or not you even like I've met people who are across the spectrum, right, who think that buckets 1 through 3 should never be done under any circumstances, and people who think that all buckets should be done at all, for any reason, and that's completely fine as well. But I think by and large, at least it's understandable, even if depending on your view on it. But it's nevertheless understandable that these are like quite clear areas. I think where it becomes complicated is with bucket two. Broadly, we've described this category as preventative genome editing. And so what does that mean? So we're talking about things that may be, for example, medically relevant and may provide a medical benefit. So something like heart disease, if you have the gene PCSK9 turned off, you're at a much lower risk of heart disease, which is, a major killer. And so that might be seen as a therapeutic intervention. Same thing with BRCA1, as was discussed earlier. If you have one healthy copy, but one mutated copy of BRCA1 and it puts you at risk for breast cancer, there's an argument for intervention there. Now, of course, it's important to remember that just because you have a faulty copy of BRCA1 or just because you have PCSK9 turned on, it's not necessarily the case that you will develop the corresponding health condition. And so there's a risk benefit ratio, whereas like if you have DMD, you definitely have a life-threatening illness. And so there's already, it becomes a little bit blurrier on terms of, is it worth doing this? And also to remember that, of course, we can turn PCSK9 off, reduce heart disease risk, but what are the other consequences of losing PCSK9, just like CCR5 with the CRISPR babies? But then we run into other issues which are even more complicated, like lifelong disabilities like deafness or developmental disorders like Down syndrome, where we start to think about, okay, well, these are conditions and impairments that may lead to, in some cases, a lower quality of life, but in other cases come with a huge amount of complications in the realm of cultural affects. Things like sign language, for example, are developed, in the deaf community, and there's a real desire to preserve them. And so, what does it mean to edit deafness? And where do we draw the line and say that one thing is normal and one thing isn't? Or where is that line? And is this really a therapeutic intervention at that point, or is it something more cosmetic? And so that you can see how that line starts to become much blurrier between bucket one and bucket two, or bucket two and bucket three. And I think that's really where the hard questions start to come in on what and where CRISPR should be used in this manner. So on that point, and I think it's important to really think about this, who makes the decisions? And this is something that is not decided. It is something that I think, and that's why these kinds of talks and these kinds of engagements are so important, because it means that you all have just as much a right to talk about this as anyone else. We want people from all walks of life to be involved in this conversation. Unfortunately, what we see, and this is an analysis I did a couple of years ago for an article at Science for the People, I looked at two of the committees that are trying to establish guidelines on the use of CRISPR, particularly in the context of heritable genome editing. So this is genome editing, which will be passed on to future generations. And what we saw when I looked at the scientists involved in these panels at the Royal Society and at the World Health Organization is that most of the scientists hail from the global north, from OECD countries. And I think that really has some questionable downstream effects, right? We have to think about, even in the case of COVID, which, this happened, this was published just as the pandemic was unfolding. But I think, we saw during the pandemic the issues with access to vaccines being limited to the global north and to communities that had more money and more access, and that had real consequences on people's health. And so when we see that this is the same sort of phenomenon happening in the context of these committees, I think we should question that. And also, I also just want to flag that if you've never heard of the CRISPR groups at the World Health Organization and the World Society that are making these decisions, they're meant to be interfacing with you, right? You're the stakeholders who need to be interfacing with this. And the fact that you maybe have never heard of them is, I think, speaks to how insular this conversation has been, but how much it needs to be expanded out. AJ, if you want to add anything.
Yeah, just to I guess reemphasize that point about how these decisions are being dominated by OECD countries and countries of the global north. And, that presents a significant wealth, racial and ethnic disparity, when it comes to decision making, both in terms of access to life-saving medication, but also the very necessary debates that need to be had around the enhancement of the human condition, whether that's preventative or cosmetic. So, as you've touched on already, it's important to highlight not just how these decisions are made, but who gets to make them.
Absolutely. And yeah, I think we often think, even if we restrain CRISPR use to just therapeutic intervention, just cancer treatments, even then, who gets to make the decision on these and where those medications are available is questionable. And if it extends into something like, bucket 3, and we start talking about things like eugenics and the specter of eugenics, if you have the global north making those decisions on behalf of the global south, you run into some real concerning areas, which I'm sure you can fill in the gaps and think about that. The other thing to think about as well is who's being invited to these conversations, not just who's on these committees, but what stakeholders are being involved. There are some people who think that if you're deaf, of course you would want to not be deaf, or if you have some sort of impairment that you necessarily would want that quote unquote fixed. But not everyone wants to be fixed, and some people don't see that as being fixed at all. A good example of this, and I think it's one that really resonated with me when I first read about it, is that historically, when looking at things like developmental disorders and intellectual disabilities, there was an overdiagnosis of people in the Black community in America with intellectual disabilities, and that had to do with things like IQ tests and intrinsic biases in those tests, which would ascribe these conditions to people, again, disproportionately in one community. Now, fortunately, when a lot of this was being done, CRISPR wasn't around. But you can imagine if you pair that bias with something like CRISPR, you start to say, okay, well, now we need to edit, but just this particular community disproportionately in pursuit of some sort of normal that we've established based on hegemonic views. And of course, in the Jewish community, we can see where this might go. We have unfortunately plenty of historical context to see where this could be a real troubling situation. So I think it's just important to think about these things and think about what we consider normal, and also where our own biases come in as people who are perhaps not deaf or don't have some of these impairments that we might see as something that should be corrected, but maybe others do not, and for good reason. So it's worth
thinking. I guess to sort of put it another way, what's the line between editing a disease versus editing your identity, right? And and that's the debate that as a society, we need to be having, and as many people need to be part of that conversation as possible. It shouldn't just be scientists in a room somewhere coming up, or clinicians coming up with the answers to these questions.
Exactly. It's very well put. And if anybody is interested in reading more about this was mentioned in the introduction, but we wrote a chapter together in 2017, as well as a follow-on article in 2019. And you'll notice that in between these two dates was the CRISPR baby event. So it was interesting to write that chapter, and then very shortly after be faced with the reality that exactly what we predicted would happen happened. And then have to respond to it in 2019 and say, perhaps there wasn't enough teeth behind our claims that we wanted a more, when I say we, the scientific community, this claim of a moratorium. Well, Dr. Hay went ahead and did it anyway. So how can we maybe address that in the future? So that's sort of what we talked about there. These are, I think, behind paywalls, unfortunately, but if anybody wants them, please reach out, and I'm happy to share PDFs of these papers. There's also an article I wrote based on some of the work AJ and I have done in Science for the People about this issue, more in the realm of regulation and some of those governance questions. And with that, I'd just like to thank everyone for your time and attention. Thank you so much.
Thank you.
Thank you. Thank you so much. That was absolutely fascinating. And I look forward to very much hearing more from the Q&A at the end. And I can see Q&A questions coming up very quickly. But before we get to that, I'd like to now hand over to Rabbi Kenigsberg, who will give his perspective from the point of view of Jewish law.
Hey, thank you, Juliet. Thank you to Gideon and Nicole and everybody at Genetics for organizing this event, for inviting me. It's wonderful to be here. And thank you, of course, to Soren and AJ for an absolutely fascinating talk to begin the evening. I'm going to be talking a little bit from the Jewish perspective. What are some of the unique ethical concerns from the perspective of Jewish law? And I think it's important to say at first that this is not a question here of, you probably all realize, but this is not a question of yes or no. Is there something that Jewish law accepts we can use or can't use? It's rather a question of how and when, and looking at that model of the three buckets. I think that gives us a good a paradigm as well. We talk about the Jewish perspective. This is obviously a fascinating technology. It has immense potential, and I would say that the highest value when it comes to Jewish medical ethics, maybe in Jewish law in general, is what we call pikuach nefesh, the ability to save a life. We're dealing with a brand new technology which has untold potential for saving life in ways that could not be done before. And that's clear and obvious, therefore, that's a tool that we need to embrace. Having said that, as we've already heard, not every application, in fact, many applications of this technology are not going to be life-saving. And that's where the questions are going to come up, and that's where things are going to get a little bit sticky. I think maybe two primary questions which we need to look at is, number one, from a risk point of view, at what point are we going to say, I think this already came up in the chat, in the questions, at what point is it safe enough to use that Jewish law will say, yes, we can go with this. This is no longer experimental. This is not that, the risks are beyond that which we're willing to take on. I'll touch a little bit more on that a bit later on. And the second question, of course, is which applications, in which circumstances, which one of those buckets, and how do we define what belongs in which bucket? But before I talk about those two things, I want to raise two issues which you may think are issues, you may think are Jewish ethical concerns, but are actually not going to be ethical concerns so much, and I'll explain. So the first one, I think, and this came up when we were preparing and talking about the session, is the question of playing God, so to speak. We hear this gene editing, we hear, it sounds very daunting, it sounds very intimidating. Is this a case here where we are playing God? Is that something, I think intuitively we feel uncomfortable with that? We think that may be a problem. So I would say that once we get to the point, this relates to the issue I mentioned already, but once we get to the point where this technology is accepted medically, where we believe that we have crossed the threshold of acceptable risk, we know enough about what we're doing, there's no reason to see this as intrinsically different from any other medical procedure. In the same way that we give antibiotics, in the same way we're comfortable with surgery, same way we're comfortable with assisted reproductive technologies, vaccines, all sorts of medical interventions. And over there, we don't say, this is playing God and this is forbidden. So too here, I don't think that we would say the same thing. The Talmud actually has a fascinating piece about this. The Talmud quotes a verse from the Torah that says, "V'rapo yirapeh," which means, it's a little bit difficult to translate. It's generally translated in English as "you shall surely heal." In Hebrew, you have the double verb form, but it's l'rapeh is to heal, and that verb is used twice. Now, the plain meaning of that verse, I'm not going to go into now, that Torah is talking in a different context. But the Talmud extrapolates from there that when the Torah tells us, "you shall surely heal," that license has been given to the physician to heal. And the question is, why do we need a specific verse to tell us a specific teaching that we have license to a physician to heal? We don't find that we're told we have license to engage in agriculture, we have license to engage in commerce, we have license to engage in architecture, engineering, anything else we do in the world. But when it comes to medical intervention specifically, we have a passage in the Talmud telling us we have license. We can interfere, we can intervene, in a sense. And there are a number of different interpretations given why that should be. But one of them is because we might have thought that exactly this, it's not our place necessarily to interfere in the world when it comes to the realm of healing, of medicine. Even today, there is so much that we still don't know. And you might think this is for God. This is not for us to interfere in that way. But no, we are taught that that's exactly, we are meant to be. There's a phrase which comes up again and again in Jewish teachings, which is partners in creation. We view ourselves that we have the obligation and we have the responsibility to become partners in creation together with God. So to the contrary, it's not that playing God in that sense is a negative thing. It's actually a positive thing. It's a positive charge that we have. And therefore, if we have the tools at our disposal, we have the science, we have the technology, again, in a way that's done that safely and responsibly, that is something which Jewish law and Jewish thinking would very much encourage us to use. Having said that, obviously, there are limits to this. And so in a narrow sense and in a broader sense as well. In a narrow sense, a fascinating example, Rabbi Waldenberg, who is was a contemporary authority on medical ethics in many areas of Jewish law. He was the adjudicator for Shaarei Tzedek Hospital in Jerusalem. And he writes, for example, when it comes to the question of cosmetic surgery, which I'm not going to get into all the ins and outs, and I will say there are those who disagree with his point of view, but his point of view was that this license to heal would not apply to cosmetic surgery. There is license for medical intervention when it is necessary to heal. But when it comes to, again, going back to those things which fit into bucket 3 or so, perhaps that license is not given. That's in a narrow sense. In a broader sense, we find as well there is an expression in the Talmud, and again, without getting into now all the technical details, but we do find a place, we do find a story, and the language that's used, I'll say it in the original language, and then I'll translate. It says, which essentially means there are things and there are considerations which are beyond us. There are considerations which are for God and not for human beings to be involved in. So when it comes to just healing the sick, that is our consideration. But thinking further down the road, thinking about aesthetics and thinking into, we get into eugenics and designer babies and all these sorts of questions. There, it's not so clear cut. But as a general overarching principle, should we be involved? Should we intervene? Yes, we should. Number one. Number two, I just want to mention very briefly that one of the ethical concerns that's raised about this is the question of autonomy. And particularly if we're talking about germline editing, that which is going to affect future generations, a question is raised: can you affect the genes? Can you edit the genes of your children, of your descendants, maybe further down the line, when you've not been able to ask them for consent? And it may surprise you to hear that this is not, again, when we're dealing with those things which are clear-cut, medical, certainly life-saving interventions, autonomy here does not play a role at all in Jewish thinking. That is something where we feel that we would have the responsibility to improve health in that way, and whether the child has been born yet or has the ability to consent. In that sense, that would not be a concern. What are the concerns? Just maybe very briefly. Two concerns that I've already mentioned, but just to go a little bit deeper. So number one, as I said, the level of risk. Is it safe? We're still at a very, very early stage in this. Was mentioned in the previous talk as well. There are concerns about off-target effects. You edit the gene in one place, you don't know what the other things that you're doing. Particularly if we're dealing with things where alternative treatments are available that we know to be safe and effective, then it would be much more of a concern. I think Jewish law would say that we would need to reach the threshold where it is the generally accepted view of the medical establishment. For this case, I think the Jewish view would take the professional view, that once it's generally accepted, this is safe, that this is effective, then it would be something we would allow. Again, there's always going to be a certain level of risk, but in Jewish thinking, it's the risk needs to be weighed up against the benefits. And clearly the lower, the greater level of benefit is going to allow us to go over a higher level of risk as well, but that threshold needs to be reached. And I think it's still very, very early days. And in many of these applications, we've not quite reached that threshold yet. Second question, of course, and this is really the million-dollar question. And I'm sorry to disappoint, but I'm not going to give you any easy answers, in the next 3 minutes. But essentially, those lines between those buckets are very, very sketchy. It's not clear necessarily. We've said something which is very much a medical intervention, life-saving treatment. You're preventing a debilitating disease. That is clearly an appropriate application of this. Jewish law would say the same. But where it is a non-medical need, and sometimes it's not clear, those things that are in bucket 2, those things between bucket 1 and 2 and 2 and 3, we have a precedent for this, or the discussion that's taken place in recent years when it comes to assisted reproductive technologies, things like PGD, pre-implantation genetic diagnosis, whereby there the questions are raised. If you have embryos in a lab and you're able to choose certain ones and not others, how far do we go? How far do we go? We can take those which have very clearly defined diseases which are going to cause a great level of suffering and possibly even death. So clearly there, the Jewish decides to say, PGD is that's an acceptable use of technology. Where it is choosing the sex of the baby, choosing eye color and hair color or intelligence, all sorts of things, there it's not so clear, and there these lines get very easily blurred. And that's where we're going to have to be very, very difficult. I think it will not come as a surprise to you when I echo the words of Soren and AJ, that there need to be different stakeholders involved in this discussion. I think Jewish tradition has a lot to contribute, and I think in two directions. Number one is that based on the age-old Jewish wisdom, based on the sources that we have, and based on the thinking that we have, that can contribute to this discussion. I think also to the converse, it's very important that the Jewish community, that faith communities in general, are involved in these discussions, because what can happen, and what has happened in other contexts, is where faith communities are not involved in the discussion, that can cause a very adversarial response. And it can be that, when people feel and communities feel that something is being forced down their throat and they have not had a say in it, they will necessarily reject that, and that's not somewhere where we want to go. So I think to conclude, this is very exciting. We embrace the possibility to heal and embrace the possibility to reach new medical treatments and interventions that were impossible in the past. Obviously, I think that the Jewish voice is very important in this conversation, that engagement with the public, with faith communities, but it all needs to be gradual. It needs to be with caution. Not everything that can be done, not everything we have the ability and the technology to do should be done. Exactly where we draw that line, that's where the caution needs to come, and that's what needs rigorous debate, public policy. But in the end, I think that we'll get there. And these things are changing all the time. So what's today may not be acceptable at a later stage, may be acceptable, recommended, even obligatory when we do cross that threshold of safety and efficacy. So those are just some thoughts.
Oh, thank you, Rabbi Kenigsberg. That was really fascinating, and I love the way you wove so deftly in with AJ and Soren's talk and those buckets as well. That was really really impressive. Thank you. Thanks to all our speakers who have given us a different and fascinating perspective on CRISPR Cas9, the future versus the slippery slope. So I'm going to hand over to Gideon Schwartz, the Genetics Education Outreach Executive, for the Q&A session for the final part of tonight's event. Thank you.
Thank you very much to Julia and to AJ and Soren and Rabbi Kenigsberg. If you have any questions you want to ask out loud, feel free to raise your hand or write in the chat. If you want to ask anonymously, you can message me directly if you want. But to start, this is probably mostly aimed at AJ and Soren, but also where would the ethical considerations stand use of CRISPR on carriers of fatal bucket 1 diseases? Could it be used to eradicate a disease prior to any suffering? And would that be a good thing to do?
AJ, do you want to take this one?
Yeah, sorry. Could you repeat that? I didn't quite catch it.
Where would the ethical considerations stand to use the use on CRISPR of carriers of fatal bucket 1 diseases? Could this be used to eradicate diseases prior to any suffering, and should it be?
Yeah, I mean, I guess that sort of speaks to that bucket 2 element that we were talking about in terms of, is it something that's preventative, and sort of defining how far we go down that track of preventing a disease. So if someone is a carrier of a disease that could potentially be passed on, is it right? Is it ethical? Is it moral that their mutation doesn't get carried to their offspring? And here we have an interesting sort of nuance. We didn't get into it during the presentation of what we define as somatic versus germline editing. We didn't get into it in too much detail. But that's where that debate happens. So somatic editing, meaning editing that affects the individual, the person themselves, and then germline editing referring to editing that will affect future generations. And I think as of today, there is a lot more comfort around somatic genome editing, so editing that affects the individual rather than germline. But this is a debate that we all need to be having, right? And thinking about in terms of what types of diseases might be appropriate and what types of diseases might not. Soren, I don't know if you have anything to add to that.
Well, just to elaborate slightly on that, in our chapter, we actually what we did in that was, it was kind of interesting. We started in the 1980s, way before CRISPR, and we talked about how gene therapies had been, regulated. So under the Carter administration, there was this report, I think it was called Splicing Life, where they were, a lot of regulation was essentially being established, or guidelines were being established on the somatic germline barrier of, if you're editing a skin cell or a hair cell or or I should say like a neuron or a heart cell, etc., that's somatic. But if you're editing a sperm or an egg cell, that's germline, and that's sort of where the no-go zone is. Of course, that barrier has been sort of, made blurry over time, because now we can turn skin cells into stem cells and turn those stem cells into egg cells or sperm cells. So, what is somatic and what is germline is a real question. But that question of future generations was a real one, and the consent of future generations. And I appreciate, I thought the rabbi's, words were very, very, very interesting. It's interesting to know that that concern isn't so live there, but it is live in other aspects of civil society. And so, I think, it's not an easy answer. I think it's maybe a bit more, obvious to say, okay, well, Tay-Sachs isn't doing anyone any good, right? There's no benefit to that really that we're aware of. So eradicating Tay-Sachs would be very helpful, right? It's a very serious condition. It becomes harder when things start to be, more risk, but not necessarily, intrinsically going to lead to a disease, as I mentioned with BRCA1, right? If you have a faulty copy of BRCA1, you won't necessarily develop breast cancer. So is it worth the risk of CRISPR editing to do that intervention? And maybe in 5 years or 10 years, where that risk has been reduced, it is worth doing. Maybe it isn't worth doing now, as the rabbi alluded to as well, that over time these things could change. So it's a difficult question, but a good one. And I think, again, all these things need to be sort of juggled at the same time.
I think that really leads on to this other question of, are there two uses of CRISPR? One to cure in one generation for some diseases, and lack of evidence of in hereditary diseases where the condition in generation one is unpredictably multiplied in future generations. I think it's
Yeah, no, absolutely. I think it's a perfect
That is
sort
that.
of
Yes.
continuation of what we were discussing, this idea of editing the individual versus editing future generations. And I guess, biology is so complex. Everything that forms who we are today is the product of hundreds of thousands, if not millions of years of evolution. And we often as scientists can be quite reductionist and try to ascribe very kind of specific things to specific genes, to specific mutations, and think of them in isolation. But biology is intricate, and these things sort of work in tandem with one another. So, as Soren and the rabbi mentioned, what looks good in one situation could actually be deleterious in another, and you don't know what the impact of that is going to be. So I think a lot of caution and a lot of restraint would have to be placed on anything in relation to germline editing, because we just don't have enough information to make those types of modifications that will affect future generations.
And that dichotomy that you mentioned, the questioner said, the potential multiplicity of consequences of not editing something now, but also the consequences of editing in multiplicity. Perhaps you've given a whole generation a new vulnerability they weren't aware of. The classic example of this, of course, is sickle cell, which was an evolutionary adaptation to malaria. Where, perhaps that's less of a live concern because we have other ways of dealing with that now, but at one point it was providing a survival advantage. And so, what would be the consequence? I think now maybe we're confident that eliminating sickle cell would be a good thing. But that's just a good example of where these things have positive and negative connotations. So.
Yeah, and to expand on that, we touch on this in our book chapter. There is, and we touched on it in the talk. There is a diversity to the human genome, and as much as part of our survival as a species is maintaining as much of that diversity as possible. And when you start eliminating things, you start sort of restricting that diversity, and that could have huge implications moving forward. So these are all these are some of the huge debate topics that we're all grappling with. But like I said, as many voices as possible in that conversation is a good thing.
And of course, what we know about genetics is limited to who we've sequenced, and that also can have biases as well, right? So anyway, it goes on and on.
I can just jump in here for a minute.
Yeah, absolutely. Thank you.
This question, the question asked about carriers of specific diseases. Obviously we're talking about recessive mutations here. It's a very interesting question, and this is something I mentioned before, that where we have an alternative treatment that's available. So I think certainly the rabbinic establishment would be a lot more reticent at first, a lot more reluctant to go into new technologies, maybe which haven't been proven. So this is obviously something that Genetics is very involved in terms of genetic testing, before marriage, and in that way, if somebody is a carrier of the disease, it's possible to ensure that disease will not be passed on. Where it does become more of an issue is where you have in certain segments of the Jewish, particularly the ultra-Orthodox community, where you have stigmas around these issues, where somebody is a carrier. That's not necessarily going to affect if the person they marry is not a carrier of the same disease, their child will not be affected. But nonetheless, there are certain stigmas that exist. So this question has already come up when it comes to PGD. Would you be able to use PGD to ensure the child is not a carrier, to eliminate that stigma? That might be having that stigma might be an impediment for that person getting married at a later stage within certain communities. And of course, there are other ways and safer ways to avoid that than gene editing, which hasn't yet been proved, if it's education and things like what we're talking about here. So I think those are all considerations to bear in mind.
Okay, we've just got time for a couple more. What work has been done to ensure CRISPR is safe and specific in correcting DNA?
I can maybe tackle that one. Although you were the head of sequencing at Desgens. maybe you know more, AJ. But it's an interesting question. It's a live one and an ongoing conversation. So two things I'll just say briefly about that. One is that CRISPR has an advantage over other technologies in that it is, in general, quite specific in comparison to older technologies like RNA interference and these kinds of things. But it still has specificity issues, and a lot's been done to try and improve specificity. What they'll do is they'll do an edit, and then they'll try and sequence around that edit and see, have we accidentally edited elsewhere? They'll look at similar sites in the genome and say, okay, well, this site looks like the site that we were trying to edit. Did an edit accidentally happen there? And that becomes a place that people might sequence to try and understand that. But this is why it's really important to do that QC analysis that we talked about. And I think it's important also to think about how therapies work, right? So we have, one of the ways we can ameliorate this risk is by performing what are called ex vivo therapies, where you take a cell from a patient or from a donor and you edit it, and then you can QC that cell and really make sure everything's okay before you put it back in. A very different kind of therapy would be an in vivo therapy. So if you ever see these terms, this is what they're referring to, generally speaking. In vivo would be to edit directly in the person, and that becomes a much harder thing to control. How do you make sure that you're editing where you want to edit? Are you editing in breast tissue? Are you editing in the liver? Are you editing in that place? And there are ways to do that, but it's difficult. And also, how can you do that quality control because you've already done the edit, right? So it's an interesting one. So I would say that those are sort of the considerations I would say around the safety, but it's ongoing. The other way we've sort of adjusted for this is there's this new technology called base editing, which is based on CRISPR as well, where there's less cutting and more changing letters. And being able to change those letters potentially, although it still has its own issues with off-target editing, maybe a more specific way of performing these, and less invasive as well, because you're, instead of cutting the DNA, which is a kind of DNA damage and particularly not good, potentially not good for the cell, you're just changing letters, which is a substitution and a bit easier going. But yes, these are ongoing concerns, and you have to balance that against what you're trying to solve with regard to the medical intervention.
Thank you very much. I think we've got time for one final question. Let's go to Simon Sandboard.
Sorry, is that me with my hand up?
Yes.
Yeah, great. Thanks very much. Just want to add, if I may, comment on the safety issue. And just to give a little bit of context, come to this from the point of view of, I run a team of genetic epidemiologists. So we look at this from a population level about how these diseases, what the prevalence of these various diseases are, and what the future health and the economic burden is, and which interventions are going to be most effective. And I think, listening to what the rabbi said and the speaker said and some of the thread in the chat, everyone's sort of looking for a fairly straightforward answer. Is this safe? And I think one thing that really needs to be brought onto the agenda here is the timescales. You often don't know the consequences of these types of therapies or any new therapy until many, many years have passed. So we do our best when we're doing forecasting to say, well, what are the assumptions and what are the risk factors involved in this? But the fact is you won't know until some time in the future. That, therefore, I think, has a bearing on, again, what the speakers have expressed so eloquently, that maybe there is an argument at this point for just restricting it to bucket one, where, you know, there's not only these are life-threatening issues, but again, to use the example of the the famous incident of the Chinese researcher that edited a baby, that wasn't necessarily just a life changing or something to mitigate the disease, but I think the big controversy there was there were alternatives, so you didn't need to use CRISPR in that particular sense. And that's what I think caused all the controversy. So I think it's again, agree with the speakers, it's tremendously exciting, but the genome and everything like that, the more we uncover it, the more we see how complicated it is, how many the interdependencies are, and you change one little part of it, you really don't know what the ramifications are, and we're not going to know for some time. So yeah, personally, I think the view would be enthralled with the excitement of this, but probably gear more towards bucket one at this stage of the technology.
Okay. Thank you, Simon. That was thanks for giving that insight. That was great. So I just want to finish off really by thanking Soren and AJ for giving us an incredible insight into CRISPR, how it's evolved over, say, the last 10 years and how it could potentially shape the future of genetics. Also, a special mention to Rabbi Kenigsberg, who shared the halakhic perspective of genetic engineering and has given us lots to think about. And an added mention, because apparently where he is, he's 2 hours ahead, so it's pushing 11:30. So thank you, Rabbi Kenigsberg, for bearing with us. And of course, to our very own Juliet Harris, who chaired tonight's event so eloquently. So thank you, Juliet, and to Gideon for organising tonight. Thank you to everyone. And the final thanks to everyone here for joining tonight's conversation. This is just another in our incredible series. Don't miss the next one, which is going to be about mitochondrial DNA, so I am told. And the date confirmation will be sent out very shortly. So thank you to everybody. Have a good evening and Shabbat Shalom.